度有一個問題，就問阿陳志偉教授嘅。大部分嗰個 vaccine 咧都係，尤其是個 messenger RNA 就 against spike protein 嘅，但係好似你個 PD-1 嘅 vaccine 咧就係 against 個 receptor binding domain 嘅。你可唔可以講下你點解會選擇呢樣嘢咧
？There's several reasons we only choose the receptor binding domain. And actually, receptor binding domain is part of the overall spike protein, which interacts with the receptor ACE2. So majority of the very potent neutralization antibody is blocking the interaction between the receptor binding domain and the receptor interaction, basically, right? So the reason we choose receptor binding domain is because before the COVID study, we actually did a SARS study, and we noticed that some of the antibody induced, if you use the complete spike, may have some side effects, right? So I still don't know whether this side effect is related to what I've already introduced during the clinical study and also based on the publication. We still do not know. But recently, we try to map all the receptor, actually all the antibody reactive domain in terms of different type of the antibody response during the natural course of the SARS-CoV-2 infection. So what we found actually among the severe patients, they do develop some antibodies against a region outside the receptor binding domain. But what's its implication? We are still doing research right now. So we still don't know whether those antibodies will cause more side effects or not. Whether this kind of side effect is similar to what we see during the SARS, right? And those are still under investigation. So overall, I think the RBD already serves the purpose to induce the most potent neutralization antibody. If we can avoid potential antibody which associate some type of side effect, that's why we choose RBD as the major immunogen design for the vaccine.